from, at the time, Research Center for Biotechnology, LIPI, and then continuous by Dr. Hendrik Winarno from Badan. And later I was involved in this collaboration. Of course, now the three labs, Professor Patumon labs, Dr. Hendrik's laboratory, and my labs were already integrated in our new institution, BRIN. In the last 5 years, this research collaboration related to bioprospecting with Fukuyama University has merged with a regional collaboration program, namely Japan, ASEAN Science, Technology and Innovation Platform, calling by JASTIP, which has initiated and driven by Kyoto University. Yeah, here also Professor Hiroshi Kamitakara also attend this meeting. Professor Hiroshi is the coordinator for this program now, especially in World Package 3 with the topic of biodiversity and bioprospecting. Before Dr. Maihara delivers his presentation, I would like to briefly introduce the JASTIP collaboration program to all of you. Allow me a share screen, just, yeah, this is just a brief introduction. Yeah, okay. So, okay, sorry. Yeah, this is the program of Japan ASEAN Science and Technology and Innovation Platform. This is a multilateral research cooperation start from 2015. 2015 until 2020 is the first phase of this project, and then continue from 2020 to 2024 for the second phase. This project was supported by Japan Science and Technology, GST. Yeah. In this project, we already create a joint laboratory between Kyoto University and at the time still LIPI. Yeah, LIPI. Two research centers involved in this collaboration. First is Research Center for Biology and also a Research Center for Biomaterial, LIPI. In this picture, we can see the activity of the joint laboratory. Our project is specific in research in World Package 3. Yeah, that conducted research on the biodiversity, especially Indonesian biodiversity in exploration or bioprospecting and monitoring, and also conservation aspect, including in this topic. Yeah. JASTIP program have two schemes. One is the core program, that is our topic in World Program 3. And the second scheme is JASTIPnet. JASTIPnet. Yeah, this is our both of scheme of our research topics. Yeah, in the center is the core project of JASTIP in World Package 3. This project title is Bioprospecting of Plant and Microbial Resources in Southeast Asia to produce highly value added product. There's three institutions just collaborate in this first phase. Kyoto University, BRIN, but at the time it's still LIPI, and then Fukuyama University. And also we have the JustTipNet project that include with Kasetsart University in Thailand and Yangon University in Myanmar. In the second phase of JustTip projects, two institutions has already joined to our groups. One is, first is Nagasaki University, and the second is National Institute of Infectious Disease, Japan. Yeah, this is now for the second phase of this project. Yeah, our new research center for pharmaceutical ingredient and traditional medicine, BRIN, will take responsibility to this research collaboration until 2024. Okay, that is just briefly introduction for our research collaboration with our partner in Japan. Okay. Save our time this morning, Dr. Mahara will present some results of our research collaborations, which is focused on the interaction of the cinchona plant, tea plants, and curcuma plant with their antimicrobial endophytes. Dr. Mahara, are you ready for your talk?
Yes, okay.
Okay. Okay, please start your talk. Now, time is yours.
Okay. Sharing. Okay. Now sharing. Microphone okay? No problem?
Okay.
Okay, start. Thank
Please.
you very much. Thank you very much, Professor Andrea, introduced me and invited in today's webinar. Terima kasih banyak. I would like to thank BRIN and for inviting me to this webinar. My name is Shoji Maehara, belong to the Faculty of Pharmaceutical Science of Fukuyama University. Today, I would like to introduce my research subject, the ability of endophytic microbe. I will focus on the production and bioconversion ability. And this presentation title is Bioprospecting Endophyte associated with Indonesian medicinal plant, valuable bioresources for pharmaceutical integrants. Especially, I investigated about Indonesian medicinal plants and their endophytic fungi. And I have proved a method to acquire medicinal components without cutting plants. This method is believed to protect ecosystems and indirectly prevent genetic disease and new pandemics. Therefore, it is necessary to understand the current ecosystem. I would like to share some of the results of my studies and research with my Indonesian friend here today to explore future forms of drug discovery. There are various elements in the ecosystems that make up the global element, and even if we consider ecosystems based on animals, including human, plant, and microbes. There are many other factors that involved in maintaining our health. For example, there are food, medicines. Food is vegetable and fruits like mango and durian. Medicine is herbal medicines and drugs brought from plants. From microorganisms, there are intestinal flora, enteral, and normal flora, and antibiotics. These ecosystems provide us with good quality water and air environment. In this ecosystem, we focus on the symbiotic relationship plant and microorganisms. So we study endophytes, which are symbiotic microorganisms of plants. Endophyte is a mixed term of two words. End means inside, phyte means plant. Endophytic microbes live within plant tissue without harm host plant. They believe that endophytic microbes have some ability. One is to protect host plant from insects and herbivores animals by producing small molecules that predator target dislike, and protect against infection with pathogenic microbes, and the host plants possess the resistance ability to the environment stress, such as dry and cold places. Okay. We focus another endophytic ability to produce secondary metabolite from the primary metabolite. This idea is important for environmental sustainability in the pharmaceutical industry in the future. In the case of our research on Cinchona plant from Chibodes, West Java, Indonesia, it is widely known quinine and quinidine, cinchona alkaloids, the chemical components, since being isolation by French researcher Pelletier and Carbantu 200 years ago. Cinchona alkaloids have been considered to be secondary metabolite of cinchona plants. However, if endophytic microbes can produce cinchona alkaloids, such as quinine and quinidine, this definition will be improved to new idea. The research procedure is research method is very easy and convenient and not so many money and so very reasonable. So we begin with separation, and at first separation of endophytic microbes using the host plant Cinchona regeriana. The young branch was surface sterilized with 70 ethanol aqueous and sodium hypochlorite 5.3%. The grown fiber from the cut face, like this, was isolated to new solid medium like a PDA, potato dextrose agar. In this case, totally 21 endophytic filamentous fungi were isolated. It was identified by analyzing the nucleotide sequence of these internal transcript spacer, ITS, including 5.S ribosomal DNA. As a result of the identification, 21 endophytes were classified into six genera, 9 homophysis species and 6 geoporte, and 2 schizophyllum and 2 penicillium, and 1 Formitopsis and 1 Alcinium. We use these endophytes to cultivation for producing second metabolite. Second metabolite is quinone alkaloids, like this. Because we want to know, can endophyte produce quinone alkaloids? So we test these endophytes from young branch producing the quinone alkaloids or not. These isolated endophytic fungi were cultured in a liquid medium, and then the culture was separated using an ion exchange resin. The removal of the solvent provided the quinone alkaloid fraction, which was analyzed by HPLC. This HPLC profile of the bioproduct produced by the endophytic fungi, CLFJ, geoporte species, CLFM, this also geoporte species, and CLFU, Alcinium species. We could confirm the endophytic fungi isolated from quinone plant produces 4 quinone alkaloids, quinine, quinidine, synchronidine, and synchronine, 4 alkaloids. We can confirm it. Next, we want to know, will quinone alkaloid producing microorganisms be passed on to the next generation along with seeds? We need isolate endophytic fungi from quinone legeriana seeds. But quinone seeds were difficult to isolate because they are very small and thin and flat. This problem was solved by using the water absorption we used during germination for seed germination. After swelling the seeds by water absorption, the phase was lightly sterilized by alcohol to separate the endophyte. When we presented to our work at the academic conference, many people involved in seed and seedling breeder asked us whether microorganisms existed in the seeds. We answered that symbiotic microorganisms exist in seeds. And so we have isolated the microbes many times and have success in isolating microbes that different from those present on the surface, different genera. So next, we need to can endophyte isolated from seeds produce quinone alkaloids. So fruits, isolate, endophyte. These endophyte culture content quinone alkaloid. Although the pH of the medium required for production was different, the production experiment was successful, and we are able to prove the production of quinone alkaloids, ClS1 and ClS4. This was also proven by LCMAS, which is a different analysis instrument from the HPLC described above. What about the plant that germinate and grow the seeds? Next question. After germination and breeding of several hundred seeds, one strain was successfully grown. This picture. This strain is adapted to the Japanese climate change in indoor environment. Endophytes were isolated from this strain. The genus of the endophytes obtained was identified and compared to those grown in Indonesia. The Japanese endophytes were of the same genus, such as Diaporte and Penicillium, same genus, Penicillium and Diaporte. However, the number of isolates was small, here is small, 8 strains. And the composition of the genera was different. We decided to investigate whether endophyte with different compositions have similar abilities. The endophytes isolated in Japan were cultivated for the production of quinone alkaloids in the same method. As a result, the endophytes of Diaporte, Penicillium, and Melier were found to be ability of producing quinone alkaloids. These results proved that the endophyte with the ability to produce quinone alkaloids can be passed on the next generation together with seeds. However, we were concerned that the amount of alkaloid producing endophytes isolated from Japan and Indonesia compare. As can be compared in the graph of production, the amount of endophyte produced from Japanese plant was less than that of Indonesian endophytic product, very small. However, it was found that the culture conditions necessary for the production of endophyte were the same as those for seed endophyte without adjusted pH. This suggests that endophyte with quinone alkaloid producing ability were passed from seeds to the next generation. So what influenced the amount, this amount difference of production? Among the quinone plants, the Legeriana strain has a very high quinine content. So we hypothesized that the quinone alkaloid content in the plant may be the cause we compare the content of kinkon alkaloid grown in Japan and Indonesia kinkona plant compare. As a result, it became clear that there was a large difference total kinkona alkaloids and kinin content in kinin content among kinkona plants of the same species, depending on the cultivation environment, Japan and in Indonesia. This is an interesting finding that the cultivation environment affects the content of plant component. The data also suggests that the different endophyte compositions along with the cultivation environment also affect the kinkona alkaloid content. In summary, endophyte taken over by seeds are involved in kinkona alkaloid production like this. The environment-dependent endophytes differ between Japan and Indonesia, different content composition. As the environment-dependent endophytes seem to be involved in the production amount, this may affect not only the amount of endophyte production, but also their content in plants. Okay, next. Next, I would like to explain shortly about the bioconversion study Indonesia medicinal plant. We try bioconversion using the secondary metabolite as a substrate for enhancement of activity and reduction of side effect and addition of new activity. At first, I thought endophyte isolated from kinkona pubescens were used for microbial conversion, bioconversion reaction using kinkona alkaloids as a substrate. The result, we were able to obtain a convert production oxygen coordinated to the nitrogen atom and one position over kinkona alkaloid. This compound was found to have antimalarial activity equivalent to quinine with improved water solubility. Okay, next. Next, it is a typical bioconversion which was carried out by Dr. Professor Andrier in Fukuyama University. Catechins have been isolated from tea leaves, tea plant, and they are well known as free radical scavengers. The microbial conversion of catechins by endophytic fungi, which was isolated from the tea plant Camellia sinensis, was investigated. As a result of this research, endophytic fungi, Geoporte species, isolated from tea plant, oxidized here catechin, which was natural type. Natural type is R configuration, C1 position, 2, 3, 4, 6 dihydroxyraban derivatives. However, minus catechin, this is a natural type, is not reacted. And this reaction is 45 years. Finally, the conversion reaction using curcumin as a substrate was carried out by endophyte associated with curcuma longa. Curcuma rhizome is used as a spice and traditional medicine. The result gave 4 colorless transformations. These were the conversions that retained the structure of phenol, kept phenol. The key function, there are key function group for antioxidant activity. This conversion product using endophytes such as these can be obtained in high yields. Therefore, it is easy to supply for medicine. Okay. In Indonesia, there are many medicinal plants, and accordingly, there are many endophytes. Recent studies have also shown that the composition of endophyte also changes depending on the cultivation environment. We believe that different reaction can be expected for each type of endophyte, and believe that the industrial applications are possible by using the production and bioconversion with endophyte described here. These result in the friendly relationship with Indonesia are based on the research and field work of Professor Shibuya and Professor Ohashi. I would like to thank these professors and all the friends involved in Indonesia, and continue this work to develop more. Thank you very much. Arigatou gozaimasu. Terima kasih banyak.
Okay. Thank you very much, Dr. Maihara, for the very interesting presentation. Here we already hear about how the endophytic microbes isolated from cinchona plants that they can produce the very valuable compound for anti-malaria, the quinine alkaloid, cinchona alkaloid, quinine, cinchonidine, and their friends. Also, we already see about endophytic microbe can pass to the next generation via the plant seeds. Yeah. Also, Dr. Maihara already isolated the interesting microbe from the cinchona plant seeds. Yeah. And one very important point that actually we already know about environment where the medicinal plants can grow up. The environment has a very significant influence to the active compound. Yeah, but Dr. Maihara also showed to us the composition of endophytic fungi also have a significant influence to affect to the bioactive compound of the host plants. Yeah. Okay. Now we will have a discussion session. If you have a question or something, please raise hand, or you can write in the chat room. Okay, we open the first session for this discussion. Please deliver your question.
Prof. Andrea, there is a question in the chat room. Probably you can see there is a question from Pak Wem.
Yeah, you can deliver your question directly, and also you can write in the chat room. This is one question in the chat room from Pak Wempi. Yeah. The question is, there is cinchona alkaloid was produced from fruits and seeds. How about the leaf? Would you mind explaining me about the production of the cinchona alkaloid from the leaf? Yeah, maybe this question, the cinchona alkaloid was also produced from cinchona leaf. Please, Maihara-san, please explain.
Yes, leaf is maybe endophytic fungus from leaf also produce cinchona alkaloids, but I not tested now. So all of parts of endophytic fungus can produce cinchona alkaloid, I think. Now I tried next generation sequencer, and the plant have what kind of fungus. So their result maybe clear that thing. Thank you very much.
Okay, yeah. If we work with endophytic microbes, we should underline which part in the host plant that produce very high active compound. In the case of cinchona plant, the main production of quinine, anti-malaria quinine, produced from the bark of cinchona plant. Yeah, not from the leaf. Maybe, yeah, already said by Dr. Marhara, maybe endophytes that living in the plant and leaf, maybe also produce a cinchona alkaloid, but maybe not so high as well as in twig or seeds of cinchona plants, because the highest production of cinchona alkaloid is in bark and seeds. Maybe that is very interesting question. Yeah, maybe next time Dr. Mahra will isolate also endophytic microbe from the leaf. Maybe, I don't know.
Yes.
Okay, we move to the next question. There is two questions from Pa Bhakti Samsu.
Mm-hmm.
Yeah, the question is Bahasa. You can answer in Bahasa. No, I can translate. Okay.
Please translate. Okay.
There is an endophytic microbe found in the cinchona plants can be categorized as a normal flora.
Normal flora. Oh.
Normal flora in the host plant.
That is Normal flora. Yes.
Yeah,
the
maybe
flora is yes, okay. Flora
maybe,
idea. Flora
maybe
idea. Yeah. Okay. You talk to me.
Maybe it's mean the endophytic microbe can produce a disease or not. If they produce disease, maybe not normal.
Yes.
But if no produce disease, maybe normal.
Yes.
What about this question? Answer of this question.
Yes, that is a very interesting question. I think plant condition depend on the plant condition. If weak plant condition, the endophytic is attack like a bite plant, but health condition is good. Plant health condition is good. The endophytic is not bright, and so symbiotic together. So both idea is good. Normal flora and intestinal flora. Both idea is good for depend on the plant condition, I think.
Okay, thank you. The second question from Pak Bhakti. How about the prospect of the how about the quality of the medicinal plants that contain endophytic fungi that can produce a specific bioactive compound in the host plant, or that same species of medicinal plant, but they have no relationship with endophytic fungi that produce a special compound. Which one is better?
What is special and new?
For example,
active.
For example, for the cinchona plants, which one is better? Cinchona plant that symbiosis with endophytic fungi that produce cinchona alkaloid, or the cinchona plants that have no contain endophytic fungi that produce a cinchona alkaloid. Which one is better?
I think for cinchona plant, existing cinchona alkaloid is better for plant because the compound is relationship of constant to composition of endophytic, so need to end cinchona alkaloids, maybe, I think. And another, if nothing, cinchona alkaloids cannot live in their plant. Their endophyte cannot live in the plant, I think.
Yeah, I think so. Yeah, maybe just now we already saw about Maihara Sensei presentation that compare the cinchona plant cultivated in Indonesia and Japan. Yeah, cinchona plants that cultivated in Indonesia and Japan, they have a very different endophytic composition.
Yes.
And also the cinchona plants also contain very, very different cinchona alkaloid.
Yeah.
That's been the cinchona plant that symbiosis with endophytic fungi that produce cinchona alkaloid is better than they have no symbiosis relationship, I think.
Yes.
Yeah, maybe already answered the question from Pa Bhakti, but Pa Bhakti also asking me, how about the research of endophytic fungi in Indonesia? Yeah. Also, moderator have
question.
Question. Okay, I can just short explanation. We also conduct the research about endophytic fungi that can produce specific active compound. Yeah, we so far already isolated many kind of endophytic fungi from many kind of medicinal plant. Yeah, now our research is still running, but we already got some powerful compound. For example, we got one powerful compound for antibiotic from endophytic fungi that associated with gambir plant, Uncaria gambir. Yeah, we isolate one endophytic fungi that produce anthraquinone metabolite that also active against many kind of pathogenic bacteria, including Mycobacterium tuberculosis. Yeah, but now is still doing the research in our laboratory. That is my question about my answer for question from Pak Bhakti Samsul. Okay, there is any question from Bapak, Ibu, please deliver your question. Yeah, this is also one question from Pak Khaidir. Yeah, Pak Khaidir now on the way. He cannot deliver his question directly. Yeah, I will read his question. Many times we found isolated endophytic fungi. At the first time, they can produce typical active metabolite. But after maintained in the laboratory for several months, for example, the ability to produce the active compound will decrease.
Yes.
You have a strategy to resolve this problem?
Yes. The ability of the production ability is reduced together with time passed. So we, after collect isolated endophytes, soon freezing -80 degree refrigerator After that, if reduce the production ability, pick up from -80 refrigerator. So their fungus is kept the producing activity. And if cannot save refrigerator, if cannot, so isolation from plant again, it is another method, two-way.
Mm-hmm. Okay, thank you very much for explanation, Dr. Maihara. I remember how to trigger the cinchona alkaloid production from endophytic microbe that isolated from cinchona plants. Many years need to perform that method. Okay. Okay. Also, there is one question from our director, Ibu Yuli.
Mm-hmm. Yes.
Actually, which one's more efficient to explore the new novel active compound, directly extraction from plant, or it should be isolated its endophyte microbes?
Oh, yes. In the future, should be isolated from endophytic because using microbes is without cut plant. So if only discover new compound is important to plant research is also very important. But in the future must be changed to the endophytic research, I think.
Okay, I agree. Maybe this is correlated with plant conservation, maybe, or maybe lands change to the agriculture.
Yes.
Yeah. If we use plant in the much amount of plant, we need to cultivate in a very wide area. Maybe like palm oil. Yeah, production of palm oil can take a very wide range conversion from the forest into the agricultural land. Maybe in the future we need to think how to search a bioactive compound from maybe friendly from environment.
Yes.
Okay. Yeah, but why? Bapak Ibu can deliver the question directly, but many questions in the chat room. Okay. The next question from Professor Hanafi. Actually, the bioconversion is a selective stereochemistry product compared by chemical method. Why? For conversion of curcumin, the product is many isomer. And which one is give a best activity? Thank you. Please, Dr. Maihara.
Yes. That not so different activity is for bioconversion product. there are almost same activity, but different because double bond is changed and stereo configuration is just changed. So not so different, and the activity of biological scavenger. So almost same.
Okay, yeah. Okay, I already answered the question from Professor Anafi. Yeah, maybe I think also it's the conversion reaction. It's depend the enzyme that produced by microbes. If they have a specific enzyme, the reaction can run in the specific reaction also, maybe like that. Okay, next question from Dr. Gian. Yeah, there, Dr. Maihara, thank you very much for that and interesting talk. I have several questions. Yeah. Well, the first question is, do the capability to produce alkali paste to the next generation?
Pass to the next generation. Yes.
Yeah, please.
Yes, yes.
Dr. Maihara.
Producibility, yeah.
Yeah, capability to produce quinquenal alkaloid pass to the next generation.
Capability is yes, yes. Next to pass to the next generation, and this trial I can isolate so very small kind of endophyte, but real is more many endophyte to live in the seeds. So another endophyte is have a more producing ability. So because seeds have more another endophytic, I think. But my technique is poor, so not so many endophyte is could not isolate it at that time, I think.
Okay, thank you very much for the answer, Dr. Maihara. Yeah, maybe if we conduct the isolation of endophytic fungi from plant organ, maybe only less than 10% of endophytic microbe that can be obtained.
Yeah.
Almost endophytic fungi have a very, very specific environment to grow up. So sometimes cannot grow up outside plant organ, maybe. The next question, how about the biosynthetic gene cluster of the respective endophytic fungi producing quinquenal alkaloid? Is it already characterized?
Not yet. Yeah.
Not yet.
Not yet.
Maybe next time we should.
Yeah. In Japan, in this researcher. Yes, in Shizuoka. Yeah.
Oh, I see.
I must.
Okay.
Yeah. Okay.
The next question: how about the biosynthetic of the cinchona alkaloid from plants and this endophytic fungi? Maybe biosynthetic route, maybe?
Yes.
Maybe.
Yes.
Is the same or different?
Yes. Yes, correct.
Okay, Pengkajian, please deliver directly.
Okay, thank you very much, Prof. Andrea. So my last question about the biosynthetic pathway of this endophytic fungi producing cinchona alkaloid and also the cinchona alkaloid from plant. But because I think because the biosynthetic chain cluster is not characterized yet, so maybe we only can guess about the third question.
Yes,
so
yes.
So yeah.
Yeah.
Okay.
I always about this thing. Yes. I think endophytic and plants both have a biosynthesis way. So plant have a, but original is plant, I think. The copy is endophytic. Yeah. So difficult to explain. So original biosynthesis way is a plant, and the endophytic fungi is copy plant to biosynthesis way. So endophytic fungi can produce cinchona alkaloid, I think. Thank you very much. Good question. Yes.
Okay, I see.
Thank you very much, Prof. Andrea and Dr. Maihara, for the answer.
Okay, it means there is a genetic transfer from plants to the endophytic microbes.
Yes, I think so.
Yeah, the latest report about genetic transfer from plants to their endophytic microbes are already published, maybe. But for the special for cinchona plant, not yet, maybe. Next work of Maihara Sensei.
Thank you very much.
Okay. Yeah, this is another question from Pak Bambang Srijanto. There, Mr. Soji, based on your experiences, how long does your research result like this can be applied in Japanese industries?
Yeah. Very long. So because I was young, so if now start in same research, that is the speed is more quickly. That time is I'm young, like master course student, so spend a very long time, about 10 years. So now I start the same research, so more short, maybe 1 or 2 years I can finish this research, I think. Thank you very much.
Okay, thank you very much. The answer, Maihara Sensei. Yeah, there is Pak Andy already raised hand. Please deliver your question directly. Please, Pak Andy.
Ya, halo.
Halo.
Selamat pagi. Good morning. My question is, thank you very much for opportunities. My question is there any experiences from your research in biosynthesis or herb medicinal that research about Moringa olivera atau disebut daun kelor? Moringa olivera herb medicinal research as far as you have been research about this. There's any experiences? Thank you very much.
Moringa.
Moringa olivera.
Moringa olivera.
Yeah, no experience.
Not yet.
That plant. Yeah, that is a plant, and no.
Yeah, there is a kind of medicinal plant that claim have a very high nutritional value. Yeah, now become popular in Indonesia. But in the past, that plant can be used to what do I say, in the magic, for magic. Yes. But now many people use for medicinal plant. Yeah, maybe you have no experience.
Yes, I have no experience, but yes, that plant have a endophytic fungus, so we can research that plant also, and
we can collect.
Yes, we can isolate from moringa plant endophytic fungus and some of bioconversion and bioproduction research we can maybe. Thank you.
Okay, thank you, Mr. Soji.
Okay, thank you. Yeah, as I said in the beginning, our session, we have one scheme of a JustTip project, namely JustTipNet. Yeah, maybe Pak Andy can make a research collaboration, yeah, with Japanese researcher and another ASEAN countries to conduct the research about endophytic fungi from Moringa olivera and make proposal and propose to JustTipNet. Maybe that is one opportunities to get research collaboration. Okay, thank you, Mr. Mahara. And there is still question in the chat room, yeah, from Pak Harto Widodo to Dr. Mahara. The question is the number and types of endophytic microbes in a plant are strongly influenced by the environment. Do you think the cinchona will not produce the same alkaloid when grow in the aseptic environment, for example, in tissue culture?
Yes. I try tissue culture of a cinchona plant, but some of tissue plant is very low content of cinchona alkaloids. So because the endophytic symbiosis is nothing, so the content is very low. tissue culture and endophytic is a mixed culture is very interesting, I think, but not test now. Thank you.
Okay. Yeah, maybe previously you already stated the biosynthetic pathway of the quinquina alkaloid originally maybe from the plant.
Yeah.
But the endophytic microbes associated with quinquina plant could copy the biosynthetic genes. Yeah, it means the sterile plant also can produce quinquina, but very low production. That's right?
Yes, yes. Thank you.
Okay. More question. Yeah, this is probably from Ibu Chandra, maybe. So, Mr. Soji Maihara, my question is about the fermentation or culture of the endophytes. I think this is the main challenge to assure the production of the metabolites of interest. What is the critical points according to your experiments? Do you think it is feasible to culture them in large scale?
Yes, yes. Yeah, that critical point is at 2 points. One is endophytic fungus can produce secondary metabolite of a host plant. It is a very critical point. And next is the ability is passed to the next generation together with the seeds. That is the next second critical point. Yeah. Yeah. Two found is very interesting us and to research is very exciting that time. Thank you. And possible to culture. Yeah. For larger scale is we try many times, but larger scale is very difficult. So because contaminant is easily to contaminate. So large scale is very sensitive culture need for production culture is very sensitive. So large scale is very difficult. Thank you very much.
Okay, thank you very much the answer, Dr. Maihara. Yeah, I think the upscaling cultivation is need to maybe specific technique, maybe, I think.
Yes.
Yeah. Okay. So many questions. There is another question from Bapak Ibu. Yeah, the question delivered in the chat room is already answered by Dr. Maihara. Any other question? Oh, already enough?
Okay, thank you.
Okay, if there is no more question, maybe we would like to thank very much to Dr. Maihara for his time to give a special lecture from his experience. Yeah, maybe we need to end this session. And I would like to say thank you very much for all of you. And Assalamualaikum warahmatullahi wabarakatuh. And time.
Waalaikumsalam.
Please, Ibu Listiana, time is yours now.
Okay, thank you very much, Professor Andrea, to lead such an comprehensive discussion. And also we would like to express our sincere gratitude for Dr. Maihara for an inspiring and also insightful talks. I believe here many audience here get many beneficial information regarding the potential of Indonesian natural products. And ladies and gentlemen, we are about to finish our event. But before that, we would like to remind you again about our next research talk that will be conducted on July 20. It will present Professor Yoshimasa Tanaka from Nagasaki University, Japan. He will present a topic about marine-derived microbes and molecule for drug development, basis for supporting innovative drug discovery and life sciences research at Nagasaki University. And to close the event, we would like to say thank you for your active participation during this event. We hope you enjoy our research talk today, and we are looking forward to see you again in our next research talk. Thank you and goodbye. Thank you, everyone.
Thank you very much.
Thank you, Ibu Listia, Prof. Agusta,
Terima kasih banyak.
Thank you, Sensei.
Dr. Maihara.
Thank you very much.
See you.
Thank you.
See you
Terima
next month.
kasih.
See you. Bye.
See you all.
Arigato gozaimasu.
Arigatou gozaimasu.
Arigatou gozaimasu.
Arigatou
Maihara
gozaimasu.
Sensei.
Shiman Sensei, arigatou gozaimasu.
Thank you, Maihara Sensei.
Arigatou gozaimasu.
Thank you, Maihara Sensei.
Thank you very much.
Terima kasih.
Thank you very much, Sensei.
Terima kasih. Arigatou.
Terima kasih Prof. Andrea. Tadi saya kelempar keluar tuh. Pasti jawab lagi.
Kok baru nongol ini?
Tadi masih di mobil.
Oh lah, terlalu sibuk sih.
Pas dijawab, pas kelempar keluar lagi.
Wah. Nanti
Tapi
diskusi sendiri deh. Ya, ya.
Asal mau bincang,
Halo,
lempar ya.
Sensei Maihara.
Oi.
Yes.
Nice to see you.
Thank you very much. Nice see you too.
Ya, mungkin acara ngobrol-ngobrol aja ya.
Iya.
Anunya ini di off aja. Apa namanya?
Iya. Apanya?
Tampilan ininya biar kelihatan orangnya gitu.
Tampilan. Aku udah pakai video kok.
Enggak, yang ini share screen-nya. Itu kan. Nah, ini kan bisa jadi lebih gede. Kelihatannya.
Oh iya.
Itu. Ya. Ya, silakan diskusi aja sendiri langsung.
Iya, Sensei Maihara.
Yes.
Thank you for your answer, but when you answer my question about the loss of the biosynthetic capability, I was not in the screen, not in the Zoom, because I was driving my car.
Yeah.
The connection is lost.
Hati-hati.
So, it is frequently we lost the capability, biosynthetic capability of our fungi isolate during subculture and upscaling. So you suggest we isolate again, right? From the source, yeah?
Screening. Sorry?
Mungkin nggak kedengaran kali, nggak jelas.
Ya, dengar.
Jelas. Iya, bergelombang kayaknya, bergelombang.
Yeah, maybe the signal already lost, just like biosynthetic capability of my fungi isolate.
Yeah, many times we found the same phenomena. Yeah. Many years ago.
How about your strategy, Prof. Andrea?
Yeah. I have one experience in 2006 and 2007. I make one proposal about secondary metabolite from certain endophytic fungi that produce antibiotic. When the proposal approved by the at the time, Riset, in the same time, my fungus also stop producing the active compound.
Yes.
Yeah, that
very
is
difficult. Yes.
Yeah, big problem. Yeah, big problem. But at the time, I think many aspects for the activate the biosynthetic gene in the endophytic microbes. We do not know yet what the specific chemical and environment that trigger the biosynthetic gene become there active to produce some specific compound. Yeah, at the time I think, in the wild medicinal plant in the agriculture or forest, they never use sterile water. They always collect water from the land. Yeah, they use the land water. Yeah, so that I try to use the land water to in cultivation of endophytic fungi. I think in the land water they contain many micronutrients like many metal or something. Yeah, maybe that micronutrient is very important to trigger the biosynthetic gene that responsible to produce specific metabolite. So I use the land's water, air sumur, to cultivate my endophytic fungi, and then they can produce again. Yeah, that is one of interesting experience.
Or co-culture, co-culture with other isolate, right?
Yeah.
Sometimes they push the production of secondary metabolite.
Yeah, there is also one method that can be applied. Yeah. Actually, they need the reason why they should produce a specific compound. Yeah, that is, if we know that answer, that is clear to make easy cultivation, but we do not know yet.
Yes.
I hope I can visit lab of Sensei Maihara in Japan.
Yeah. Okay.
Next year. It's okay.
Please come.
If pandemic is okay, pandemic.
And I want to go to Indonesia, to meet my friend and to eat the mango and durian.
That is very important.
Yeah.
You know, Sensei Maihara,
Yeah.
To find durian in Jakarta, there is one specific place. This is in the near of my apartment. Yeah. So you have to visit my home.
Your garden
Yes.
have a durian tree.
There are so many seller of durian.
I think it's better durian of Medan. It's a very famous ucok, durian ucok.
Medan.
And very cheap,
Yeah.
very cheap.
Cheap.
Yeah. In Medan,
Medan non.
Okay, Sensei Mahera, Professor Andri, I have to go. Thank you for the talk.
Okay,
Thank you very much.
Yeah.
Okay, thank you.
Hati-hati driving.
Ya, thank you. Bye-bye.
Bye-bye.
Pak Hendik mana? Aku diam aja.
Nggak ada dia, Pak Hendik. Halo, Pak Hendik.
Nah, itu baru keluar tuh.
Mana? Oh, itu. Hendik Sensei. Ah. Gimana, Pak Hendik?
Hendik-san?
Yes.
They come join this.
Yes.
Yeah.
He present it. And it's okay. Unmute. You. Pak Hendik? Kemana?
Nggak ada.
Ada tadi ini dia, tapi lagi mati unmute.
Iya.
Ini. Kenapa? Mungkin itu kali bergelombang kali ya.
Okay, maybe we should finish
finish
our
Mahara
meeting
Sensei.
today. Thank you very much.
Thank you very
Mahara
much.
Sensei, for your time.
Genki de.
Yes, ありがとうございます。Terima kasih, good communication. ありがとう。
Please read my SMS, WA, WhatsApp. Okay.
WhatsApp. Okay.
Yes. Okay.
Oke, terima kasih Bapak Ibu semua. Kita cukupkan sampai di sini. Ya. Mungkin bisa kita off ya, Bapak Ibu panitia. Terima kasih.